Hi everyone, my name is Sue, and today I want to tell you a little bit about an ongoing project we have in the V Synthetic Biology platform to create next-generation Cas9 activators. And so I know many of you have heard of Cas9 as this revolutionary tool that's fundamentally changing our ability to edit and manipulate genomes. But really, what is Cas9 at its core? Cas9 is simply just a protein, and it's a protein that works in concert with a small guide RNA. This sgRNA binds to the Cas9, and it programs the Cas9 to target a specific DNA sequence. When Cas9 identifies a target sequence that's complementary to the guide RNA, it binds, and it acts as a pair of molecular scissors, cutting the DNA. This tool is so powerful because it can be easily repurposed to target any genomic locus simply by swapping out a small sequence within this guiding RNA. And so naturally we asked, what else could we do with Cas9 besides cut DNA? And so we can take Cas9 and we can mutate its catalytic residues and create a dead Cas9. This dead Cas9 still binds to an appropriate target locus, but it no longer cuts the DNA because the molecular scissors have been disabled since the catalytic residues have been mutated. Instead, the Cas9 simply just binds to the DNA and it sits there like a molecular couch potato. We can then fuse that molecular couch potato to an effector protein and use this fusion in order to regulate gene expression. For example, we could use it to upregulate any gene in the human genome on command. So we and other groups were super excited about this prospect, and so we decided to take the dead Cas9 and fuse it to the most potent effector protein we could find in the literature to upregulate genes. We then targeted this fusion to a toy system consisting of a minimal promoter and a red fluorescent protein reporter gene. When we targeted this fusion to this minimal promoter, we expected to see a large amount of protein expression corresponding to essentially super bright red cells. This was our expectation, and then this was our reality. We saw next to nothing. Our fusion activator was a dud, and unfortunately, we were kind of stuck at this point. So did we give up? No way. Instead, we decided to take a cue from nature and think about how genes are naturally expressed. In nature, several expression factors cooperatively assemble at a promoter, and they work together to pull down the necessary machinery that's needed to express a gene. So we wondered whether or not we could replicate this with Cas9. So we decided to create a second-generation activator, and we did this by fusing a potent combination of 3 separate activator domains to Cas9. We then targeted that new fusion to the same toy system, and we wondered whether or not we could get potent activation of this reporter protein. Fortunately for us, we saw many red cells and really strong fluorescent reporter expression. So this was very exciting. But at this point, we had only activated gene expression from a toy system. And the true test would be if we could take the human cell line that we've been working with, and instead of activating this toy system, instead direct our activator to genes naturally found within the human genome and activate gene expression from our chromosomes. We wondered if we did this, could we activate gene expression to a significant extent? So we tried this, and one of the first targets we tried to upregulate was NeuroD1, and with our first generation activator, unfortunately, as expected, we only got about a 3-fold modest increase in gene expression. However, when we targeted our second generation activator to that same target site, we were able to induce a robust 87-fold improvement in 87-fold induction of gene expression, corresponding to a 30-fold improvement above what we were able to achieve with our first generation activator. Similarly, we were able to upregulate another gene target, ASCO1, roughly 4,600-fold above background, leading to a 40-fold improvement above our first generation activator. And then finally, we were able to upregulate expression of Titin, which is the largest protein coding gene in the human genome and cannot be overexpressed any other way. And we were able to upregulate this gene 20,000-fold above background, corresponding to a 320-fold improvement in gene expression above the first generation activator. So at this point, we were very excited because gene expression is the way Mother Nature takes stem cells, or a mass of identical cells, which are embryonic stem cells, and produces you, or I, the whole human body. This is how Mother Nature takes stem cells and directs differentiation of them into each of the different tissues that consist the human body, including neurons, heart cells, or even liver cells. So we wondered whether or not we could take a set of stem cells and upregulate gene expression to a high enough extent such that we could differentiate those stem cells directly into neurons. And so we attempted this, and we activated a gene responsible for stem cell differentiation into neurons. And this is what we expected. This is the ideal situation. We thought we would see a mixture of undifferentiated iPS or stem cells, shown in blue, along with a large number of neurons, shown in red. And as expected with our first generation activator, we couldn't really induce high enough levels of gene expression to induce or actuate this differentiation mechanism. So when we target our second generation activator to that same gene, we're able to induce much, much more robust levels of stem cell differentiation into neurons, which is very exciting. And this corresponded to a 22-fold improvement in the percentage of neurons we could produce using our second generation activator versus what we could achieve with our first generation activator. And this is very exciting, and we wanted to make sure that this tool was not only applicable to the research done in our lab, but also to research that's done in other labs as well. So we attempted to activate genes in other model organisms, including mouse cells. So we were able to activate gene expression roughly 1,200-fold in mouse cells, corresponding to a 70-fold improvement in gene expression over the first generation activator. Similarly, we were able to activate gene expression in fly cells, corresponding to 3,300-fold induction of gene expression. And this corresponded to a 300-fold improvement above the first generation activator. And then finally, we were bold, and we attempted to induce gene expression in yeast, which are removed by about 1 billion years from humans in evolutionary time, and we could still activate a gene 80-fold above background level of expression, corresponding to a 6-fold increase in gene expression above the first generation activator. And finally, we have a joke in our lab, which is that if you're doing something, you should probably try to just multiplex it. So we were wondering, could we do this with our Cas9 activators? And this is really cool with activation because multiplexing allows us to activate entire genetic networks simultaneously at once. So to test multiplexing, we targeted our Cas9 activators to 4 separate genes at once, and then we benchmarked our first generation activator against our second generation activator. Overall, we were able to induce modest levels of gene expression for the 4 genes we tried to activate with our first generation activator. But with our second generation activator, we achieved a far more robust response, overexpressing each of the 4 genes to an average of 15-fold higher level of expression across the board. So this is all really cool and leads to a bunch of immediate applications for in vitro work that we can do in our lab. And because we can activate genes on command, this now allows us to conduct functional studies of the whole human genome. Similarly, because we can induce differentiation of stem cells into neurons, this begs the question of whether or not we could produce other specialized cell types for high-value tissues. And finally, because we can multiplex gene activation, we wonder whether or not we can create and understand new and more complicated genetic circuits. And all of these different applications have obvious implications for human health and disease, particularly for regenerative medicine. Thank you. I'd like to thank our PI, George Church, for offering us this opportunity and an awesome lab space and environment that really facilitates creativity, and the rest of the activator team who really made this happen, including Alex Chavez, John Scheinman, Ben Pruitt, Marcel Tuttle, and the rest of the Church Lab. I also want to give a big shout out to the Wyss Institute for the space and the resources that providing the space and resources, and also Harvard Med School and MIT Biological Engineering, and finally NSF for funding this operation. Thank you.